Thank you all for attending. My name is Marc Johnson. I am a virologist at the University of Missouri. Until the pandemic hit, I used to be a molecular virologist that worked on HIV replication, but in pandemic times, I have become a specialist in monitoring viruses through wastewater, which is something I was not expecting. The work I'm going to show you today is a collaboration with the Missouri State Health Department and the Missouri Department of Natural Resources. And if you're not familiar, this is the state of Missouri. This is currently the places where we are sampling wastewater to look at SARS-CoV-2 levels and lineages. At this point, we're at about 102 communities, as well as 36 state facilities, which includes all of our prisons, plus some veterans' homes, psychiatric hospitals, and other sites. We have a website anyone can go to. It's live. You can look and see. We update it every week to show what's happening in terms of raw wastewater SARS-CoV-2 RNA levels. So this is just sort of will reflect what's happening in your community. The red triangles means that the RNA levels are going up, so almost 100% of the time reflects increases in patient levels in those locations. But what I'm mostly going to talk about today is our efforts in sequencing SARS-CoV-2 and the lineages through wastewater. And I have to confess that when I first started this project, I didn't think there was going to be any point in this kind of project. But I changed my mind largely after I this paper that I'm going to quickly summarize for you. It was first submitted by Jesse Bloom's lab in December of 2020. This is right around the time we were seeing the first variants appear. This is when the UK and or alpha, beta, and gamma were starting to hit the scene. But if you recall, from when SARS-CoV-2 first started its pandemic until practically the end of 2020, there was very little variation in the sequences of SARS-CoV-2. There was maybe one mutation, the D614G, which had really taken over, but there weren't a whole lot of changes that we were seeing in sequences, at least not that were seeming to really take off, which kind of surprised me because I would have expected it to adapt a bit more when it jumped into a new host, but that's what happened. So if nothing happened after the first 9 months, I wasn't necessarily expecting anything to happen. But then in this manuscript, what they did is they looked at another coronavirus that's been infecting humans for quite some time, at least 50 years, which is this called 229E. It's a coronavirus that will cause a common cold. And what they did is they looked back in time at lineages and how they changed over the decades. And there are three main points I want to highlight from this manuscript. The first thing they found, which is I find amazing, is that this virus evolves over time, but as a single worldwide lineage, which means that in 2020, the 229E in China and US and Brazil and South Africa are all going to be more similar to each other than from a 229E from 5 years earlier. It seems that there's one sort of global strain that sort of evolves together. The second thing was, and this really impressed me, is that the virus is clearly evolving resistance to our human vaccine response, our antibody response. There's no vaccine against it. And the way they found this is because they had serum from patients at different periods in time, and they could check what viruses their antibodies were able to neutralize. And what they found is serum from a certain year could block virus from that year, but if you took virus from a few years in the future, that virus will have gained resistance to those antibodies. Very clearly, the virus was evolving to adapt to the immune responses we were mounting. And then the final point was they tracked down sort of what changes were causing these resistance to the antibodies, and almost all of it tracked to this spike protein. So this graph at the bottom shows where there were mutations occurring in this gene, and this hot spot right here where you see a lot of changes was called the RBD, or the receptor binding domain. This is the part of the protein that actually binds to the host. This is what determines tropism, and this is where most neutralizing antibodies target. There are a few other places on the spike protein where antibodies can neutralize, but at least in humans, over 90% of them will target this receptor binding domain. One other thing just to point out is the spike protein has two pieces, S1 and S2, which are cleaved by a host protease, and this is also an important area of evolution. So once I read this paper, I was convinced that even if we hadn't seen SARS-CoV-2 mutating yet, I was sure it wasn't going to go away, and I was pretty sure it would start to evolve once there was herd immunity or something approaching herd immunity. So we began the project of sequencing, and again, what we focused on was the spike protein. So this is the picture everyone's seen of SARS-CoV-2. The spike protein is a red blob on the surface. This protein has two jobs. One is it binds to the host cell receptor, which is called ACE2, and the second is it facilitates penetration into the cell. So it actually causes the membrane of the cell to fuse with the membrane of the virus. Now, there are a lot of groups that have begun characterizing SARS-CoV-2 lineages through wastewater, but there are basically three approaches that can be utilized to do this. The first is using qPCR, and what you do here is you can design a qPCR probe where you only get a signal if there's a sequence that has a particular mutation in it. So you can screen for mutations that are known to be in particular lineages. These qPCRs are great. They're fast, they're quantitative, and they're fairly cheap. But they have the disadvantage that they're not really telling you about the whole virus. They're only telling you about one nucleotide position in the genome. And there is a lot of redundancy among the different lineages. So it can be misleading, and you kind of have to adapt to the virus' ever-changing landscape. The second approach is the one we use, which is a targeted sequencing. So we don't amplify the whole genome. We only amplify that region that I showed you is likely to be evolving, the receptor binding domain and a few other spots on the spike gene. And because we're not sequencing the whole genome, we're actually only amplifying a certain piece, which then we can sequence all together. So if there's multiple mutations, we can tell which mutations were derived from the same lineage, and that's really key. The disadvantage of this is it takes longer, it is more expensive, and it can be misleading in terms of quantitation. So particularly when RNA levels are really low, if patient levels are low, if you extract your wastewater and you have a tube where there's only 10 strands of RNA, and you go to amplify it and only one of those 10 amplify, you're going to be misled because you only know about that one sequence. So especially when it's low, the quantitation can be misleading. So you have to be cautious about that. But the big advantage is that it tells you about the relevant region and it tells you what things go together. So it's really a great way of seeing what new sequences are evolving. The third approach, and this is the one that most people use, is whole genome sequencing. This is basically using the same technology that is used in clinics to amplify and sequence the entire genome of the virus. So the way they do that is they break it up into little tiny pieces and then sequence them all together, and they throw it into a software pipeline that then spits out what the whole sequence was. And this is an excellent approach when you have a uniform sample, but it gets a little bit hairier when you're working with something like wastewater, where you have a mix of often many different lineages. So what you end up with is not a whole sequence, but you have basically a list of all the mutations and polymorphisms that are present. And because it's done in little bitty pieces, you can't really tell which mutations go together. This approach is still very effective at figuring out what known lineages are present, but it's not as good at determining if there's something new. Just quickly, this is our workflow. We get samples 5 days a week. We extract them. The process we use takes about 6 hours. We always quantify them the same day in which the state, which is almost immediately put onto that website I showed you. When we go into the variant detection, we do two parallel things. We run some qPCRs against 3 of the known probes, which reflect some of the known variants of concern, and we do our targeted amplification. I mostly wanted to show you these are the 3 sites that we'll target. The most important one is this RBD, receptor binding domain, but we also will sequence the N-terminal domain and the S1-S2 junction. So almost all neutralizing antibodies will hit the RBD or the NTD, And the S1, S2 tells us other things about the evolution of the virus, because often with many of the variants of concern, they have mutations right at the furin cleavage site. They're believed to enhance furin cleavage. The furin cleavage site was a recent acquisition by this virus, and it's actually suboptimal. So it seems that gaining a more efficient furin cleavage site enhances virus replication somehow. This is the second map that is on our dashboard. This one actually shows which variants we detected at the different locations. We started doing this with samples in January, and this was the first round that we published on the public dashboard. When we began, it was kind of boring. All of these brown squares means that we didn't detect any variants. It was just the original, the same strain that we'd been seeing all through 2020. We started this process just before the Alpha variant, or the UK variant, which I can't stop calling it, appeared in Missouri. On this first round, we saw a high prevalence of the UK variant in only one location, which was part of St. Louis. However, as the weeks went on, as everywhere, the Alpha variant sort of spread out through the state. What was interesting to watch is that it spread from sewershed to sewershed in a fairly predictable manner. First, it sort of filled out St. Louis and started to work down the interstates. And I should mention these yellow triangles means that there were multiple variants present. The green cross means the only variant detected was the UK variant. And so it sort of followed the interstates and went through the populace, and then eventually made its way out to all of the rural communities. And eventually by the end of April, it had fully displaced the parent lineage. We were no longer detecting the lineages we were seeing all through 2020. All through the year, like most of the U.S. in 2020, the cases were going down, and it seemed like it might be coming to an end, and then that all changed on May 10th. This was the first date when we detected the Delta variant in Missouri, and it was one of the first outbreaks in the United States, actually. The first detection was in a city called Branson, and if you aren't familiar with it, this is a real tourist destination. It's a very country western themed location, so a lot of people were traveling to and from this. At the time, this wasn't the Delta variant yet. The word Delta had not been coined yet. They hadn't come up with this whole Greek alphabet yet. It was just one of the India variants. And it wasn't even the first, it was actually the third India variant we had seen in Missouri. We had seen the two others earlier in the year. But Delta, or what it was at the time called B617.2, is the one that the World Health Organization had sort of said appears to be the most infectious version. It was not actually a variant of concern yet in the U.S. for some time. But I knew the World Health Organization's recommendation, so it really had my attention. And sure enough, this is just 14 days later, the next at a collection, and you could see that the variant had just gone scattershot across the whole state. And unlike the Alpha variant, it didn't follow the interstates. It just sort of went and appeared everywhere. So immediately we were seeing it in rural communities throughout the state and not just in big populous areas. The term Delta was actually coined on May 31st, but it wasn't named a variant of concern until mid-June, by which point it was practically the only lineage present in our state. This was June 14th. This was the last week or the last sampling, I believe, when we detected a sample where there was no Delta. This was the latest reading from last week. These turquoise mean Delta, so it was the only thing we were detecting. Just want to show you what the output looks like for me. One of the tricks with doing any kind of PCR-based sequencing is PCR can do funny things to your sequence. There are sometimes errors, there are sometimes recombinations, and you have to be able to sort out what's real and what's not. We published our software platform recently, and we have a few different software methods we use for assembling the data and sort of pulling out and removing anything that appears to be likely a PCR chimera or polymorphism. And this is what my output looks like. It comes out as a spreadsheet of, here's what we see, L452R, T478K, if you're not familiar, that is the Delta variant. In this case, it was a little over 70%. Second one here was N501Y, A570D, which is the Alpha variant, or the UK variant. And frequently we'll see a lot of extra polymorphism here and there. Sometimes this is just a PCR error, sometimes not. But fortunately, you can always look it up. So, like, this is one I've seen many times. It's not even an amino acid change. It's a silent mutation at amino acid 574 of the spike gene. But I looked it up. You can look on this thing called GISAID, where you can blast and see any sequence. And sure enough, this is a real mutation seen in patients, and it seems to be enriched in Missouri. But it's cool. It's just to demonstrate we see not only sort of the raw lineage, but new subvariants that can go with it. Now, this slide is just to remind you that we aren't just sequencing a few sites. We are collectively sequencing about 150 different sites. So what I'm going to show you next, I can say is unusual because it's something we did not see very many of those 150 locations. So I'm going to tell you about this one sewer shed in Missouri where when we first started sampling, all we detected was what I call here is other. So this pie chart just shows the distribution of the sequences we pulled out from the wastewater. In January, there was nothing but the original strain, but in February is when the Alpha variant first moved in. So on February 22nd, we started to see this B117, which is Alpha UK, has many names. No surprises there. But then something a little surprising happened. The first week of March, we detected a new lineage which had a common mutation, which is L452R. This had already been seen many times in Missouri. It's found in the Epsilon, or the California variant, and it's also one of the mutations that's present in the India variant, such as Delta. But this was not Delta, and it was not the California variant. It was something else. And it also had another polymorphism, which was N460K, which is a uncommon but not unheard of polymorphism in spike. But what really hit me is the week after, when we started to see some more polymorphisms appear, and they weren't just anywhere. They always were in the backbone of this lineage that had L452R. We still had some of the 2020 strain, we still had UK, but we had these new lineages. And the part that really freaked me out was this, is that on the second week we saw N460K, but we saw it twice. We had two different nucleotide changes at the same position in the virus, which both led to the same amino acid change, which is really flies in your face that there is some kind of strong positive selection going on here, that it really wanted to have this amino acid change. The next week, again, started to see more of the variants, and more polymorphisms were appearing with time. After about 4 weeks, we started to see these new ones, N439 and N440, and some more as well. So this is just this lineage is just to summarize that this was not one single lineage. It was a bunch of different. It was like a quasi-species. They all had. It was all made up of these same 9 polymorphisms, but they had different combinations of them. And yeah, it was really weird. I had never seen this kind of evolution from any sewer shed with this virus. Usually, you'll see 1 or 2, and that's it. Here we were seeing scores. So I did a lot of digging to try and figure out what was going on with these mutations, and this got me really excited. This is another manuscript from Jesse Bloom's lab, where they took the SARS-CoV-2 spike protein and they made every possible single amino acid change in the spike protein, and they tested them all to see whether they had gained resistance to any neutralizing antibodies. And in this case, they were using the 2 neutralizing antibody used in the Eli Lilly monoclonal therapy. So I mentioned before, over 90% of neutralizing antibodies target the SARS-CoV-2 spike, and they're broken into 3 or 4 classes, depending upon how they bind to spike. And antibodies within the same class are often affected by mutations at the same location. So a mutation that blocks one class 1 neutralizing antibody is likely to block multiple. And what jumped out, so the way this looks is shows the amino acids shown are ones that cause resistance to these 2 neutralizing antibodies, bamlanivimab or atezolizumab. And what pointed out is these main mutations that we were seeing off the bat were very very much enriched in their ability to neutralize these monoclonal antibodies. If you know, Regeneron is the other company that has these monoclonal therapies, and theirs they have a class 1 and class 3 antibody. And so those positions that were coming up late, those 439 and 440, those were the only ones that did not cause resistance to the ELI. They do cause resistance to Regeneron. So pretty much every one of these amino acid changes we've seen were either affecting the receptor binding domain properties or it was affecting neutralization. So at this point in time, what I thought was happening is we were seeing resistance within people that were receiving these therapies, but I no longer know if that is true. I suspect it is not. So I'm a virology lab, so one of the cool things we can do is recreate some of these sequences in the lab and make what's called pseudoparticles and test their properties. So one of the things we did is tested whether they are indeed resistant to these neutralizing antibodies, and shown here, so the way this looks, every dot here represents an infection, and from left to right we add more and more antibody. So this red line in the top left graph is the wild-type virus, which I call D614G. As you can see, as you add more and more neutralizing antibody, you'll see less and less viral infection. Whereas all three of the mutants we're testing here, which are all combinations we saw from the Missouri Sewershed, have resistance to class 1. They all have resistance to class 2 as well, but only one of them has resistance to class 3, which is the one that has the N439K. So everything behaved exactly as you would expect based on its genotype. We also did some whole genome sequencing. What I was looking for here is I was suspecting that this virus had mutation in its proofreading enzyme. It just seemed that it was mutating too fast. So I wanted to see if there are any mutations in other parts of the genome which might cause it to be something that was mutating faster than normal, but we found no such thing. We also learned that it's really hard to tease out whole genome sequences from this approach when it's a mixed sample. We know that this sewershed only had alpha, gamma, or V117 and P1, and our oddball lineage, but it was impossible to try and tease out what the 3 different full-length lineages actually were. So it's just really hard to tease out a whole sequence when it's a mixture of such complex sequences. So moving on, almost as surprising as its arrival was the departure. By the end of April, the frequency of these oddball sequences started to go away, and by the beginning of May, they were gone. I've sequenced the Sewershed every week since March. We have not seen a single lick of these sequences again since. And I should mention, they have never been seen in a patient. Of the 2 million sequences in GISAID from around the world, none of them have sequences that are even close to what we were seeing from this Sewershed. So while we were finding these oddball lineages in Missouri, and I was really getting excited of what the heck is going on, I reached out to just about everyone I could think of who was doing wastewater surveillance across the country to ask if they were seeing similar things. Is this just a Missouri thing? And they all said the same thing, which is, we don't know. We're doing whole genome sequencing. We can't really tell if there are new lineages. But then I was listening to my favorite podcast, which is called TWIV, and they had some guests on from New York City who were actually using the same approach that I was of amplifying and sequencing the RBD. So I reached out to them and we had a chat to see whether we were seeing similar things, and they basically said, we are seeing oddball polymorphisms, but we can't really tell if they go together because they hadn't really worked out the software yet, and they also were amplifying a much smaller piece of the receptor binding domain. So we decided to collaborate, and they sent me almost the next day RNA from all 14 of the Sewersheds representing all of the boroughs of New York City, and we sequenced them all, and it was really cool. It was like a who's who of all of the variants of concern. But there are also a handful of lineages that did not match any of the known lineages that you would find in the database. They were expressed in very low amounts, and they did not match the sequences we were seeing in Missouri, but they were very clearly unique. We went and did the same thing 2 weeks later, and we found exactly the same exact things were still present. It wasn't some sequencing thing. These were present in particular Sewersheds, and they stayed geographically constrained. And interestingly, they were going up in frequency, it seemed. In retrospect, we're pretty sure what's happening is the amount of these oddball sequences were remaining constant, but the amount of patient RNA at this point in time was going down as there were fewer and fewer patients. But it didn't last, as we all know. So this is a summary of 3 of the sewersheds and the lineages we saw from them. This is all just showing the receptor binding domain. One of the things that really jumped out at us is that all of the lineages from New York, they were very divergent from one another, but they all had a mutation at position 498. And in particular, a lot of them had the mutation 498Y, or glutamine to tyrosine. Now, if you look on GISAID, of the 2 million sequences, this particular change, which actually takes 2 nucleotide changes to make this 1 amino acid change, has never been seen in a patient. However, it has been seen in adapted models. So a guy named Ralph Barrick, if familiar, was the first to make a mouse-adapted SARS-CoV-2, and one of the mutations he made for making that was Q498Y. So this is when at first the light came on. It's like, oh, maybe these are not coming from humans. These could be infecting some other animal. I also want to point out this one from Wastewater Treatment Facility 3. It's just crazy. This receptor binding domain has 16 amino acid changes, including a deletion of position 484, which is a really key one for evading class 2 neutralizing antibodies. And yet we recreated it, and it was still fully functional. This is a real sequence that's circulating somewhere. One thing we wanted to see is if it actually has gained the ability to infect other animals. So again, we generated the sequences into our SARS-CoV-2 pseudoparticles, and we tested different cell lines that express different ACE2. So they could all infect cells that have human ACE2, but the red bar is the parent sequence, or the D614G. As you can see in the cell lines that have mouse or rat ACE2, you can see that the red bar is very low because it cannot utilize those receptors. The blue bar is N501Y, A570D. That's the alpha variant, which is known to have gained the ability to infect rodent cells. And you can see they all have enhanced infectivity in the mouse and rat ACE2 cell lines. And all of the mutations all of the variants we pulled out from New York City Sewershed had indeed gained the ability to infect cells with mouse or rat ACE2. It doesn't mean that that's where they came from, but it at least is consistent with the hypothesis that this might be coming from rodents. We also tested these guys against neutralizing antibodies, and like in Missouri, they all behaved exactly as their genotype would predict. Some of them were resistant to all 3 classes. We also tested their neutralization with serum from actual patients, and in general, they were resistant. The patient serum could still neutralize, but usually at a lower level. So we still don't know where these sequences are coming from, but the fact that each sequence has remained constrained to a particular sewershed for about 8 months now really does not sound to me like it's coming from humans. We think it's coming from some animal that is geographically constrained. So one of the things we've done is we sequenced the ribosomal RNA from our same sewershed samples to see what animals are contributing to the wastewater. And mostly what we found is what you would expect, which was we found human ribosomal RNA, and we find the thing that humans eat, because of course those would make it into the sewershed as well. So we saw a lot of cow, pig, sheep, goat, chicken, things we eat. But the things that probably could be something else, or the ones that are our likely candidates right now, are cat, dog, and rat, which were all fairly frequently present in the sewershed. One of the other things we've been doing is amplifying larger pieces and sequencing it using a platform called PacBio. This has been a collaboration with the University of Wisconsin and the CDC. It's pretty cool that you can see this big piece like that. This was one sewershed where there were only 2 lineages. The first one was our oddball lineage, and you can see it. This little square around the RBD is the part we were seeing before, but when you see the whole S1 region, you can see there's lots of big deletions and a bunch of other changes. But the second lineage was very clearly had all of the polymorphisms consistent with B.1.621, which I believe is now called Mu. I think it's the latest one that's been added to the Greek alphabet. So this is another technology that is coming forward, which could really shed light on variants within a sewershed. So that's my last slide. So we submitted a manuscript with the New York City work about 3 or 4 weeks ago, and about 2 or 3 days after we submitted the manuscript, the USDA also submitted a manuscript. And in their case, what they were doing is surveillance of white-tailed deer, and lo and behold, they checked their serum and found that they very frequently have high levels of antibodies against SARS-CoV-2, which almost certainly means they have been infected. So the finding that SARS-CoV-2 is in other animals is not particularly surprising anymore. So let's remind you, this was a collaboration between University of Missouri, the Department of Health, and the Department of Natural Resources. It was actually the Department of Natural Resources that started this project. And with that, I thank you for your time.